Hello everyone, this is Dr. Divya. And today's topic for discussion is sudden blotting technique. In this video, we are going to discuss about introduction to blotting techniques, transfer process, blotting membranes, types of blotting techniques, methods and applications. Definition for blotting: it's a molecular technique of transferring proteins, DNA or RNA onto a carrier or membrane. This is done after a gel electrophoresis, transferring the molecules from the gel onto the blotting membrane or adding the samples directly onto the membrane. Types of blotting is classified into three major groups. Sudden blotting, it is used for separation or detection of DNA. Second one, northern blotting, it is used for separation or detection of RNA. And western blotting, this is used for the separation or detection of protein. Transferring process of blotting technique. Here we used three types of method. First type is capillary method. It is also known as wet transfer method. In this, the biomolecules are transferred from the gel to the membrane using the capillary transfer method. Second type, electroblotting. It relies upon current and a transfer buffer solution to drive protein or nucleic acids onto a membrane. Third type, vacuum blot method. In a vacuum blot, instead of using capillary force, the force that will transfer the DNA is a vacuum. The membrane used in blotting technique, three types of membrane we used. First one is nitrocellulose, nylon membrane, aminobenzyl oxymethyl cellulosic membranes. And these are all the membranes used for blotting techniques. Sudden blotting. And this method involves separation, transfer and hybridization. This technique was developed by E.M. Sudden in the year 1975. The sudden blot is used to detect the presence of a particular piece of DNA in a sample. The DNA detected can be a single gene or it can be part of a large piece of DNA such as a viral genome. The key to this method is hybridization. What is hybridization? It is a process of forming a double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target DNA. Methods involving in sudden blotting. First, the restriction endonuclease cut high molecular weight DNA strands into smaller fragments. The mixture of fragmented DNAs is separated in agarose gel electrophoresis. The restriction fragments present in the gel and denatured with alkali and transferred onto a nitrocellulose filter or nylon membrane by blotting. Then, this procedure preserves the distribution of the fragment in the gel, creating a replica of the gel on the filter. A sheet of nitrocellulose or nylon membrane is placed on top of the gel. Pressure is applied evenly to the gel to ensure good and even contact between gel and membrane. Membrane is then baked in a vacuum or regular oven at 80 degrees Celsius for 2 hours or exposed to the ultraviolet radiation to permanently attach the transferred DNA to the membrane. Now, the probe is added to the matrix to bind the molecules. Any unbound probe are then removed. The place where the probe is connected corresponds to the location of the immobilized target molecule. Applications of sudden blotting. It's used to identify specific DNA sequences in DNA sample. It is also used for isolate desired DNA for construction of rDNA. It is used to identify for the mutation, deletion and gene rearrangements. In genetically modified organisms, it's used for testing to ensure that a particular section of DNA of known genetic sequence has been successfully incorporated into the genome of the host organism. It is also used for phylogenetic analysis. It is used to determine the number of copies of a particular DNA sequences. In DNA fingerprinting, it is used to identify paternity and maternity test. It is used in the forensic department and also for the personal and biological identification. And these are all the books for the references. Thank you.